Okay, so today we're finishing photosynthesis, okay, which means we're doing the second half of the reactions. Friday we did light dependent. We had to have those photons of light in order to get our reactions to occur. Today we're doing light independent reactions, okay, but let's do a little bit of review first. So first, this is our photosynthesis equation. What part of the equation did we do last time? So this one kind of evenly separates out into light independent and light dependent. So can you identify in the equation what we did last time? What was the light? Talk to your neighbor. See if you guys can come up with it. The light dependent reactions. Did it? I don't know. Okay, so what on the left side is the light dependent reaction? H2O, yes, right? Because what did we do with H2O? We split it. We took off electrons and protons, and then what was left over? Oxygen, right? Okay, so there's our light dependent reaction. Okay, so today then we're moving on to the light independent reaction. And this part doesn't require light. And we're taking carbon dioxide and we're fixing it. We're making it into organic molecules that can be later used for energy. Okay, and what is a molecule that we identified as having energy in it? Glucose, right? Okay, so this second part is carbon dioxide, converting it, running through a cycle called the Calvin cycle, and running through multiple carbon dioxides to make sugars. Okay, so one thing in here, what's not shown in the reaction in our equation is our products, right? So what were the products of the light-dependent reaction? Right, NADPH and ATP, right? And also oxygen, right? Okay, and these are going to be used in our light-independent reactions. And so they will be used and converted back into ADP and NADP+. And then they can be sent back to the light independent reactions to be used again. What's that? So ATP, when we go through chemiosmosis, we get ATP. Say again?
In the intersection of the diagram?
No. It's actually in chemiosmosis, which we didn't. It's not in the diagram. Yeah. We create the proton gradient, which then we use to make ATP. Yeah. Okay? All right, so we'll talk now. What I want to do next, though, is a little review from last time. So you guys remember the acronym? You all got that right on the quiz, right? Okay. All right, so what does PPC, PPFN stand for? Okay, can you guys remember? And what I would do is write it out. Oops. So the first P stands for photosystem dose. All right, and the second P is?
PQ. I can't remember.
Good, plastiquinone. Yeah, if you say it in a British accent, it's better. Plastiquinone. See? Rolls off the tongue. When I looked up how to pronounce that, it was like the British pronunciation. So.
Yeah.
Plastiquinone. All right, what's the C? Cytochrome. Complex. All right, what's the next P? Getting ahead of yourself. Cyanin, yeah. Plasticyanin, okay. Do you know what cyanobacteria are? Yeah, they're greenish blue bacteria that can photosynthesize, so plasticyanin. It's referring to that bluish green.
Is there any at the end of it?
I don't think so, but we'll put one on anyway.
Just to make out
All right. I'm going to take it off. All right, what's the second P, or the fourth P, really? This is everyone's favorite. We're just a little disappointed that he came this late in the game. All right, then F, ferredoxin. And then the last one, okay. It's a lot of big words. They're huge. Okay, so now can you make a Z diagram? Can you put them in the right order? It doesn't really, it actually makes more like an M, but it zigs and it zags. All right, what would be a good place to split these two if you're going to draw one half and then the other? What's that? Yeah, so if you split it right here, one on the left makes one hump of the M, the other one makes the other hump. All right, cytochrome complex. Photosystem 2 uses light to excite the electron. It's accepted by plastoquinone, which then donates it to the cytochrome complex, which donates it to plasticyanin, which donates it to photosystem 1, which uses light again to excite the electron again, accepted by ferredoxin, and then by NADP reductase. Okay, that's nuts and bolts. All right, what detail would you add to this? Antenna complex? Yeah, you could add that. What'd you say? At the end? The NADP to the NADPH? Is that what you're saying? Yeah. Okay, good. All right, so at the end, NADP is reduced to NADPH, and that's the final fate of that electron. It's added on to NADP+. Okay, what else? What's at the very beginning? All right, so you put water here. And you get 2 electrons. You get 2 protons. And you get oxygen, right? All right, what else? How about, we have some protons here. We have another source of protons. What's that?
I'm saying, isn't there chemiosmosis in there?
Okay, that's at the end, so we won't put it on this, but yes, those are used for chemiosmosis later. But something else contributes to the proton pool. What's that? No, that's where it comes from. It comes from those hydrogens, yes, but there's another proton being pumped through one of these things.
cytochrome complex?
Right, so cytochrome complex also takes that energy to pump protons.
So close. Yeah, it's so close.
Okay, anything else? I think that's a good... You could draw it all out with the proteins embedded into the membrane and then show chemiosmosis. But this, I think, is a good starting point. Okay. What's that?
That's too much work.
Well, it's a good idea. Figure it out. All right, so you guys got this down? Okay, all right, go to a blank one and about the Calvin cycle. All right, does this look familiar?
Yeah.
What does it look like? Yeah, it just looks like the Krebs cycle. We're just going to do the opposite. Rather than releasing carbon dioxide, we're going to capture carbon dioxide. Okay? And we're going to add it to a 5-carbon molecule compound called RUBP. It actually has a much longer, more complicated name, but we're just going to use the acronym RUBP. Okay? And it has, in order to do that, it uses an enzyme called Rubisco. Okay, so it adds carbon dioxide to RUBP, and then it makes these 3 carbon molecules called 3-PGA. So the first step is carbon fixation, right? Or carboxylation, you could say, right? We had decarboxylation in the Krebs cycle. Okay? And then what we do is we convert PGA into G3P, okay, in a few reduction reactions. Okay, these are both 3 carbon molecules, but we're going to rearrange some of the bonds. Okay, and in order to do that, we need two ATPs and 1 NADPH. So we're using the ATP that we created in the light-dependent reactions. And then that G3P can be used to make sugars. All right, what was the last step of the Krebs cycle? Right, regeneration of oxaloacetate, but we're not going to regenerate that here. We're going to regenerate RUBP. And this also takes another ATP. Yes?
So I noticed it's going from the 2 ATP to the ATP, so these are breaking down the stuff and makes these up. That means this is a catabolic reaction, right?
Yes, so this fixating carbon dioxide is an anabolic reaction. Using ATP is catabolic, right? So we're using ATP, we're using that energy to add carbon dioxides together.
Gotcha. So we're using the energy we created in order to make.
Yes, exactly. Yes. So, and the way I have it written here, this is per 1 carbon dioxide, you use 2 ATPs and 1 NADPH, and then a total of 3 ATPs. But to get a 3 carbon molecule, you need to run 3 carbons through. Okay? And so to get a glucose molecule, how many carbons is that? 6. So how many carbon dioxides would you have to run through in order to get? Yeah. So you'd have to run it through 6 times. Each time you're going to add another carbon. And that would be enough to make a glucose. Yeah. Well, G3P is a 3 carbon molecule, but that's used to make sugars. Yeah.
What is the G?
It's a long word. It's like I don't know.
Some shipping.
Yeah. Glide bisphosphate or something like that. Yeah, so how many carbon dioxides would you need to run through to make sucrose? 12, right? Yeah, because it's 2 6-carbon sugars. So you run 12 carbon dioxides through. That's enough to make a sucrose. And that's what plants do. So they make sucrose, and then they send that to other parts of the plant to make it into starch by capturing carbon dioxide. And that's the total process then of photosynthesis. So we went from light and water and carbon dioxide, and we ended up with sugars and oxygen as a byproduct. Okay, any questions? Yes?
2 questions.